1A carbohydrates, monosaccharides and disaccharides. Organic compounds. Chemical compounds in which one or more atoms of carbon are covalently linked to atoms of other elements, most commonly hydrogen, oxygen, or nitrogen, less frequently nitrogen, sulfur, and phosphorus. Carbon. Normally, each carbon atom can make 4 bonds and form a tetrahedral-shaped molecule. These small units are called monomers, while linking these molecules form polymers. The ability of carbon to make macromolecules is the basis of all biological molecules. Carbohydrates. Most abundant group of organic compounds on Earth. Major elemental composition is C, H, and O. General formula. There are 3 main groups of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides. The simple sugars, they are called simple sugars. A general formula for this can be written CH2ON. All are reducing sugars. 3C-triose.C3H6O3. 5C pentose sugars. C5H10O5. Ribose, deoxyribose. 6C hexose sugar. C6H12O6 glucose, fructose, galactose. Glucose has 2 isomers, different forms: alpha glucose and beta glucose. Disaccharides, the double sugars. 2 monosaccharides join together by a condensation reaction, releasing a water molecule. The bond is known as glycosidic bonds. The double bonds formed are 1 to 4 glycosidic bonds, 1 to 6 glycosidic bonds. Sucrose. Stored in plants such as sugar cane. Glucose plus fructose. Lactose milk sugar. This is the main carbohydrate found in milk. Glucose plus galactose. Maltose malt sugar. Found in germinating seeds such as barley. Glucose plus glucose. Except sucrose, others are reducing sugars. Reducing sugars are the carbohydrates with free aldehyde and the ketone group. Reducing sugars are carbohydrates that can oxidize other substances by contributing electrons while being reduced. Practical skills testing for sugars. 1 milliliter of the analyte sample must be mixed with 2 milliliters of Benedict's reagent and heated in a bath of boiling water for 3 to 5 minutes. The development of a brick red colored precipitate of cuprous oxide confirms the presence of reducing sugars in the analyte. Heat a non-reducing sugar such as sucrose with a few drops of hydrochloric acid. Allow it to cool and then neutralize the solution with sodium hydrogen carbonate. Checkpoint. 1. What are carbohydrates? Carbohydrate is a biomolecule consisting of carbon C, hydrogen H, and oxygen O atoms, usually with a hydrogen-oxygen atom ratio of 2 to 1, as in water, and thus with the empirical formula CmH2ON. 2. Describe how a glycosidic bond is formed between 2 monosaccharides to form a disaccharide. They are sugars formed by joining 2 monosaccharides by a glycosidic bond. Glycosidic bond is formed by removal of a water molecule from two adjacent disaccharides by a condensation reaction. Water molecule is formed by removal of OH group of one monosaccharide molecule and H group of adjoining monosaccharide molecule. 3. Why is HCl added first to identify non-reducing sugars in Benedict's reaction? Break the glycosidic bond in sucrose through hydrolysis. 4. What is the color change of the reducing sugar when heated with Benedict's solution? Blue, green, yellow, orange, brick red precipitate. 5. To what monosaccharide does glucose break down in respiration? Triose, 3C.